Hello biologists and welcome to Communicable Diseases 4.1.1 taken from the OCR specification for A-level Biology A. Today we're looking at lesson objective E on phagocytosis. So there are a couple of defense mechanisms that we can have in our body. Last lesson we looked at non-specific primary defenses such as a physical barrier like the skin. And today we are looking at non-specific defenses such as phagocytosis. Next lesson, we're going to be looking at specific cell and immune response. A little bit more difficult. That's next lesson. All right, so phagocytosis obviously involves a phagocyte. It's also known as a macrophage. And that's what it looks like. And phagocytosis, this is an outline of what's happening. If you want to pause it and try and figure out what's going on, please do so now. Otherwise, we're going to move on. So phagocytosis, this is what happens. Here you can see at step 1, a pathogen is being engulfed by a process of endocytosis. This forms a membrane-bound sac which contains the pathogen, and this is a phagosome. The phagosome then fuses with a lysosome, and don't forget lysosomes from back in cell structure. These contain hydrolytic enzymes. So this means that the hydrolytic enzymes can now access the pathogen because the lysosome has fused with the phagosome. And the lysosomes break down, or hydrolyze is a better terminology to use, hydrolyze the pathogen into harmless products, such as amino acids. Now, these amino acids can either be expelled from the cell that you can see here in step 6 in a process called exocytosis, or they can be reabsorbed back into the cytoplasm of the phagocyte and used to build its own proteins again. Also, the phagocyte will undergo something called an antigen-presenting cell. It will become an antigen-presenting cell. And it does that by taking the antigens off the surface of the pathogens, off the surface of the pathogen, and it'll present it on its own cell surface. So these antigens here that you can see on step 5 here, these have been taken from the pathogen. So it's taken the antigens from the pathogen, put it on its own cell surface membrane to become an antigen-presenting cell. And you're going to look at more about why this is so important in the next lesson objective. So we've looked at those things so far. We're going to look at the roles of cytokines and opsonins. So cytokines, these are released from T helper cells to trigger clonal expansion of the B cells. You're going to look at this in more detail in the next video. Opsonins, these bind to antigens on the cell surface of pathogens to increase the rate of phagocytosis. So an antibody acts as an opsonin because it binds, an antibody binds to an antigen on the cell surface of a pathogen. So we're going to have a look now at some blood smears and to see what we need to be able to identify in a blood smear. So here are some examples of a blood smear. We need to be able to identify a neutrophil as having a lobed nucleus. We need to be able to identify an erythrocyte, which is also known as a red blood cell. These all appear red, and you can kind of see this biconcave structure whereby the middle of the red blood cells do appear lighter in colour, giving away that biconcave structure. We have lymphocytes, which are again a type of white blood cell, which have a spherical nucleus. And then we have monocytes, which are unilobular nuclei inside. This again is a type of white blood cell. The main white blood cell we need to know about is a neutrophil, but I have seen lymphocytes and monocytes examined in a question based upon their microscope images. So that's that lesson objective, guys. Please make sure that when you are drawing an image of cells that you are using smooth continuous lines, no shading, you're labeling at least 3 things with a straight line with no arrowheads, you're filling 50% of the area, you're putting your magnification on there, your title, and some kind of annotation to describe maybe a colour of something that you can see within the image. Don't forget in your exam, don't use the words amount, it, they, or size, and good luck with your exams.